Hi, myself Rani Shri. Today I am going to give a presentation about the topic Anfinsen Experiment for protein folding. This experiment was discovered by Christian Anfinsen while he was studying the enzyme ribonuclease A. He explained about the information regarding the protein folding and he explained the sequence of the amino acid and also says that the protein has the capability of folding itself and it has also having a lowest energy conformation. In this experiment, the following features that he had explained are: firstly, the disulfide bridges are disrupted by the beta-mercaptoethanol by undergoing reduction. Nowadays, it had become an indispensable tool which are used in STS page for the purification and separation of proteins. And the information regarding the protein folding, they are resided in the amino acid sequences. So, he chose a ribonuclease A as his model in order to explain the protein folding. But he failed to explain unless that enzyme is treated with beta-mercaptoethanol, also known as 2-mercaptoethanol, and urea, so that the protein becomes unfolded. So the protein unfolded can also be refolded once he removed the urea and beta-mercaptoethanol so that the ribonuclease A regains its biological activity. However, he also explains that the removal of mercaptoethanol but not the urea leads to the 1% recovery of the activity. So, this led to the formation of a random disulfide bridges between the 8 cysteine residues in 105 different possible ways. But the 100% recovery is not possible because some of the mistakes also made in vitro condition and also in a vivo condition. So he discovered an enzyme called as protein disulfide isomerase, that is PDI, which reduces the incorrect disulfide bridges which are present on the surface of the misfolded protein. He also shows that the correct 3D conformation also results in the correct protein folding. This PDI enzyme plays an important role in protein folding and it catalyzes the incorrect disulfide bridges to be reduced and PDI itself be oxidized. This enzyme recognizes the incorrect disulfide bridges which are present on the surface of the misfolded protein. But the correctly folded protein has the disulfide bridges which are resided in the hydrophobic interior. So here there are the two pictures indicating that a native protein becomes a denatured protein and the denatured protein can be refolded. In the first photo, the two cysteine molecules are held together by the disulfide bridges. So this disulfide bridges can be disrupted by treating it with a beta-mercaptoethanol. This breaks the disulfide bridges between the two sulfur atoms, leading to the formation of two cysteine residues. And the sulfur atoms which are present in the beta-mercaptoethanol, they form a disulfide bond at the end of the two molecules of beta-mercaptoethanol. In the second photo indicates that the native folded protein undergo denaturation by reducing the disulfide bridges and adding a 8 molar urea. Then the denatured protein undergo refolding by the removal of urea and then undergo oxidation. This shows more than 90% activity of the protein. This is generally in observation. But we can control under the laboratory condition by reducing the disulfide bridges between the two sulfurs of native protein and adding a 8 molar urea to form a denatured protein. Then the denatured protein undergo first oxidation and then the removal of urea leads to the 1 to 2% activity of the protein. Coming to the conclusion of this experiment, that the Anfinsen says that 100% recovery can be made in vitro to its native conformation, that disulfide bridges cannot be reduced manually but can be done in vitro. So he also explained that the informations which are required for the protein folding, they are resided inside the amino acid sequences. Thank you for watching this video.